So in section 3, we're going to look at the step 2, which is the presentation of antigens. And our learning outcomes are to compare the terms antigen, immunogen, and epitope, and to list the characteristics of antigens that optimize their immunogenicity. Describe how the immune system responds to allergens and superantigens. So these are two different kinds of antigens that we're going to differentiate between. And list the types of cells that can act as antigen-presenting cells. So an antigen, going back to this description here, is anything that provokes an immune response in specific lymphocytes. So it's specific to it. To be perceived as an antigen or immunogen, it has to meet certain requirements. They have to be foreign, have a specific shape and size and accessibility. And so the property of behaving as an antigen is antigenicity. So not all substances provoke an immune response for whatever reason. They may not be foreign or complex or have the right size, and so that's what we're going to look at, is things that have a better immunogenicity or antigenicity. So the characteristics of antigens are that they must be perceived as foreign. They can be either whole or parts of microbes. They can be cells or substances that arise from humans, other humans, animals, plants, or other microbes. And they have to be complex, not repetitious in composition. So they usually contain proteins or protein-containing compounds like glycoproteins or lipoproteins, and those are more immunogenic, and usually that's because they are not repetitious. So if you look here, you can see different things that are good immunogens. Proteins, epitopes are the sequence or pieces of that protein that are specifically what the lymphocytes bind to, the receptors bind to. They can be from another human, like you could have A-type blood and you get B-type blood, and that can create an immune response. So it could be foreign to you. It's not from your own body, but it could be from another human. Microbes are obviously something, microbial cells or viruses, and then plant molecules. But a poor immunogen would be something like this that is repetitious, this polypeptide, even though it's protein. It may not be big enough, or it may be repetitious here because it looks like those are all the same amino acid, and then trisaccharides probably is not big enough, but it also could be that it is repetitious. So chemical categories of antigens: proteins and polypeptides. So proteins are at the top of the list. They are the most immunogenic. And then lipoproteins, glycoproteins, nucleoproteins, all of these have protein components to them. These things are more dense and more regarded and usually bigger, but they're not complex. I mean, they are not repetitious. They're usually complex. And so these are going to be more of good immunogen. And polysaccharides and lipopolysaccharides, especially like lipopolysaccharides, are something that we don't see in humans typically. Like lipopolysaccharides are something found in gram-negative bacteria in the outer membrane. Some polysaccharides, but not all, depends on whether they're repetitious or not. So like starch, probably not it is not an immunogen because it is repetitious. It's all repeating subunits. And cellulose, not an antigen. Even though it's very long, it's all repeating glucose, so it's not complex. So the effects of size, they have to be large enough to catch the attention of surveillance cells to initiate an immune response. So generally, you have to have at least 1,000. Very seldom do we have antigens that are less than 1,000. And if you have 1,000 to 10,000, this is molecular weight, so the measurement would be Daltons, or atomic mass units. 1,000 to 10,000 are weak antigens, and then the most immunogenic are like 100,000. And typically these are very large, long proteins, polypeptides. So think of a protein, again, is typically about 100 or more amino acids. And think about the size of those amino acid molecules. That is very large. And so if you have like glucose, for example, glucose is C6H12O6. So let's think about this. So let me write this out. you have C6H12O6. This is glucose. Carbon is typically 12, and so we have 6 of those, so that would be 72. Hydrogen has a mass of 1, and you have 12 of those, so that's another 12. And oxygen has a mass of 16. You can look that up in the periodic table, and there are 6 of those, so that is 96. I'm doing my math right. So this would be 10 and 8, yeah, 180. So it's 180 Daltons, or molecular weight. And so is glucose antigenic? Is glucose something that our body, our immune system is going to recognize as foreign? So if you answered no, you would be correct. So the epitope, as we're going to see, is a small portion of the antigen. It's not the whole antigen, it's just a part of the antigen. So here's an animation on that.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
So as you can see, epitopes are different than an antigen. Antigen is the whole, and the epitope is the portion of the antigen that is recognized and responded to by a lymphocyte. And it is the part of the molecule that is identified as foreign. It is the primary signal that says, hey, this is foreign, it's not supposed to be here. So haptens are something that consist of only a determinant group, the epitope itself. And they're too small to be seen as dangerous, so they do not elicit an immune response by themselves. But if they are linked to a carrier molecule, then the combined molecule develops immunogenicity to it, so it is immunogenic. So as you can see here, a hapten inserted into a mouse would not produce antibody, but if you put the hapten on a carrier molecule and inserted it into the mouse, it would create an immune response. But the immune response is not to the bigger portion, the part that is the carrier molecule, but yet to the antigen, the hapten itself. And so the hapten is still what is giving you the response. So an example of a hapten is penicillin. So penicillin, some people are allergic to it, some people are not. The idea is that possibly hapten, that the penicillin is a hapten, so in some people it's not going to elicit an immune response, but in others it combines with a carrier molecule which does make it antigenic to them, and so they are sensitive to it. An alloantigen is a protein or other molecule of one person that is antigenic to another. So it's part of one molecule. So like the blood markers in ABO blood type would be an example of an alloantigen. So that's the basis for the blood typing, and we'll talk more about this in chapter 16, but this is the basis for incompatibilities with blood typing and for organ grafting as well. So it's something that is from another human, and it's part of another human, but it's not a self-antigen, so you count it as something that's foreign. Superantigens are bacterial toxins that are really potent, and they activate T cells at 100 times greater than the ordinary antigens. It's a very non-specific binding, and it can create or release an overwhelming amount of cytokines and cause cell death. So toxic shock syndrome toxin in Staphylococcus aureus is an example of one of these. So this was just making a note on here that this is like the Rely brand of tampons, we found that they were creating these toxic shock syndrome cases in the 1980s. And they took these off the market because there were so many cases of this. But what was happening is that the applicator for the tampon was creating microabrasions within the vaginal tract. And that was allowing the bacteria to get in if they were bacteria that produced the toxic shock syndrome toxin. I have a hard time saying that. But if they had that toxin, then they would just grow and replicate. And the other part of this issue was that at the time they thought, oh, these are super absorbent and we can wear them for 18 hours. Well, you know not to do that now because of this situation that happened in the 1980s, and there's all kinds of warnings on the box about this. So the tampons were left in a long time. It's during your period, so you have a blood flow that is present. It's warm, it's moist environment, it's perfect pH, perfect conditions for out-of-control growth. And these bacteria, the Staphylococcus aureus, are beta hemolytic. So they use the blood, break it down, and release the iron that they need to grow. So they're going to be able to grow really readily in this environment. But the specific cells that cause this toxic shock syndrome, they produce the toxic shock toxin. And so not all of the staph bacteria did this, but the ones that had the ability to produce this toxin were the ones that were causing a lot of damage, and women were dying from this. So we'll show you an animation just on how the T cells get activated so readily.
Antigens are normally processed by antigen-presenting cells and presented to T helper cells on class II MHCs. Only the T helper cells with T cell receptors capable of interacting with the antigen are stimulated to produce cytokines. The activated T helper cell stimulates only B cells that react with the same antigen, thereby limiting the response to the production of antibodies that were presented. Superantigens, such as the toxic shock syndrome toxin produced by Staphylococcus aureus, are not processed by antigen-presenting cells. Instead, they bind directly to the outer portion of an MHC class II antigen on antigen-presenting cells and to the outer portion of the T cell receptor of T helper cells. Rather than binding to only the cells specific for that antigen, which represent about 1 in 10,000 T cells, the superantigen binds without antigen specificity to as many as 2% to 20% of the T cells. This results in stimulation of many T helper cells. The result is the release and activity of excessive amounts of interleukin-2, IL-2, and other cytokines that enter the bloodstream instead of only acting locally as they normally do. This leads to symptoms of fever, nausea, vomiting, diarrhea, and sometimes shock, with failure of many organ systems, circulation failure, and often death.
So we're going to go back through that a little bit. So this first section is like how you can see the antigen doesn't respond to it doesn't target and doesn't every T cell. But when it finds the T cell, it has specificity for it does produce cytokines that activates B cells, also displaying the same antigen. And that's the way it's supposed to be. It's specific to the particular antigen. But with the superantigen, you can see here, it's sort of bringing two not specific T cell and B cell, or macrophage and T cell together, and it's causing it to activate it, even though it's not specific. So they don't match. And so 2 to 20% of the T cells in your body are being activated all at once instead of the normal amount. And so it's about 0.2% that's usually what actually are being activated. And so this causes all the symptoms of the shock because of all the cytokines that are released.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So I want to point out a couple of things that have to do with this particular video. So they were showing internal protein, internal antigen, and external antigen. So in this particular part, it's showing how, like, foreign protein, this is, let's say, viral protein, made through biosynthesis. They're going through the process of making capsids and things. And self-protein also will be broken down and end up in the rough endoplasmic reticulum. And in here, they are going to be presented with the MHC1 molecule. That then travels through the Golgi apparatus. And the normal process, kind of letting the T cytotoxic cells know that this is supposed to be here, is this process where it presents self-antigen in the MHC1, and it ignores it. But if it's a foreign antigen like this from a virus spike, piece of the envelope, or nucleic acid, something is presented in this MHC1 molecule. And when the cytotoxic T cell binds to it specifically, then it's like, ah, there you are. And it's like going to cause it to activate the T cytotoxic cell, and it's going to blow the cell up. So it gets activated that way and makes many, many more T cytotoxic cells, and then goes around and looks for that particular antigen. So in the external environment, if you have a phagocytic cell that brings in a foreign particle, it eats it up, chews it up, and then complexes it with a MHC2. Notice this is a class II molecule. It goes to the surface and presents that antigen on the surface, which then will attract another kind of T cell, the T helper cell. The CD4 cell will bind to that, and that's how that gets activated. So the antigen is processed and presented, and it can be done, it can be presented through an MHC1 or an MHC2. If it's an MHC1, it's a cytotoxic T cell. If it's an MHC2 molecule that it's presenting the antigen, it's going to stimulate a CD4 cell, a T helper cell. And here's a rather lovely picture of a dendritic cell engulfing a fungal spore. So you can see the little kind of tan part, that's the dendritic cell, and then there's the fungal spore. So it's got little finger-like extensions. That's why it's called a dendritic cell, and that is an antigen-presenting cell. So our concept questions, concept check 15.3, list and describe 3 types of molecules that serve as antigens. True, false, a molecule of 1500 molecular weight is strongly antigenic. Number 3, the blank is the molecular fragment that signals that the molecule is foreign. Number 4, a blank is too small to be recognized as a foreign molecule and requires a carrier to be seen by the immune system. And number 5, T cells respond to blank at a rate 100 times greater than ordinary antigens. So question 1, we had several molecules that were serving as antigens, but mostly proteins or proteins combined with other molecules. So proteins are polypeptides. Proteins like glycoproteins, lipoproteins, nucleoproteins, any of those. And then polysaccharides can also be long polysaccharide chains if they're complex. So those are some examples of types of molecules that serve as antigens. And the true or false, a molecule of molecular weight 100 or 1,500 or 1,500 is strongly antigenic, and that is false. I wouldn't say not antigenic, but it's not strongly antigenic. Strongly antigenic would have to be 100,000 molecular weight, or atomic mass units, also known as Daltons. And so this is weakly antigenic, if it is antigenic at all. So number 3, the blank is the molecular fragment that signals that a molecule is foreign. So the epitope is the, or antigenic determinant, is the molecular fragment that signals that a molecule is foreign. That's what the lymphocytes, the receptors, respond to. A blank is too small to be seen or recognized by the foreign molecule and requires a carrier to be seen by the immune system. That would be a hapten. So question 4, it's a hapten. Question 5, a T cell responds to blank at a rate 100 times greater than ordinary antigens. So this would be the specific antigen called a superantigen, which we spent quite a while talking about. And that's the end of section 3.